This idea and manuscript were formed on the basis of the plastic pollution and persistence problem. Petroleum-based plastics is an environmental concern due to the difficulty of their degradation, and some, such as single-use plastic bags, cannot even be recycled, leading to the accumulation of this waste, which will remain for years and damage the environment in the process. As such, interest in the method to break down single-use plastics in the environment has grown. In this manuscript, the enzyme laccase of Pleurotus ostreatus is being investigated for its putative activity related to plastic degradation, based on interest and information found in multiple research papers. This manuscript utilizes secondary data, specifically the Laccases database, to collect information on the different laccases which have been found in Pleurotus ostreatus. And using the accession codes on NCBI, the different laccases are put through BLAST in order to find other laccases that have a high percentage similarity to the sequence. This is then repeated for the remaining laccases, and the fastest sequences are collected and used to generate a phylogenetic tree and align sequences using clustral omega. The phylogenetic tree with real distances was then used to derive the results. The phylogenetic tree showed that the different laccases of Pleurotus ostreatus were not monophyletic, and so these branching patterns showing distinct monophyletic groups that exist support the idea that there may be more than one copy of laccase. This is further solidified with the Laccases database showing that subspecies of Pleurotus ostreatus can have more than one type of laccase. So this diversity and multiple copies of laccases in Pleurotus ostreatus alludes to the idea that these laccases have diversified and so maybe have picked up the function of plastic degradation. Furthermore, given that there is still much that we don't know about fungi and its functions, it is reasonable to hypothesize that the plastic degrading ability currently accredited to Pleurotus ostreatus is not restricted to the species. Other Pleurotus species may have mechanisms that break down this plastic. So the laccases of the Pleurotus ostreatus species show a high similarity to the Pleurotus ostreatus, and this relatively high similarity leads to the hypothesis that laccases of other fungal species under said genus may also have the ability to degrade plastics.